Welcome again to the video table of contents for AJP-Heart and Circulatory Physiology, or From the Editor's Desk. In the March 1st issue, you'll find some outstanding pieces of work that cover a range of cardiovascular science. So here's a sample. In a study by Zucker et al., the interaction between caveolin-1 and the AT1 receptor was investigated as a mechanism that prevents angiotensin-induced constriction. In skeletal muscle resistance vessels from caveolin-1 knockout mice, repeated applications of Ang2 exhibited sustained constrictor responses in contrast to reduced responses in vessels from wild-type mice. When silencing CAV1, the amount of membrane-associated AT1 receptor was significantly reduced by short-term angiotensin exposure. The authors conclude that CAV1, through a direct interaction with AT1 receptors, delays internalization and resensitization of the AT1 receptor. Venable and co-workers applied spatial Fourier analysis to confocally recorded fluorescence emitted by a mitochondrial membrane potential dye following global myocardial ischemia. Using several interventions and pharmacological agents, the data show that the period of asystole is strongly correlated with the time of ischemic mitochondrial depolarization, which may indicate a causal relationship between mitochondrial membrane depolarization and asystole. Chen et al. Report that resveratrol activates SIRT3 in cardiac fibroblasts in vivo and in vitro. Resveratrol prevented fibroblast to myoblast transformation by inhibiting the TGF-beta SMAD3 pathway. Furthermore, resveratrol prevented the development of cardiac hypertrophy in response to hypertrophic stimuli in wild-type mice, but not in SIRT3 knockout mice. This finding provides insight into the cardiac remodeling process and potential for new therapy. Finally, in one of our rapid reports, Deo et al. Demonstrated significant differences in oxidative stress components from peripheral blood mononuclear cells in young black men compared to whites. Blacks showed elevated intracellular superoxide and increased protein expression for GP91, P47, suggesting an upregulation of the NADPH oxidase pathway in blacks. Interestingly, a positive family history of hypertension did not further enhance these parameters. Thank you again for watching this installment of Video Table of Contents Highlights. We will see you in 2 weeks, and don't forget, send us your best work now.